I'm going to, whoops. And I'm going to turn my screen. Now, sometimes when I go to present mode, my screen locks up. So if it locks up on your end, let me know, because I can't always tell on my end. So if somebody could turn on their audio and let me know if they're seeing the first question on their screen. Is that what you're seeing?
Yeah.
Yeah? Okay, thank you. All right, so the first question on this assessment was, there was 2 experiments that were described. They both had to do with yeast and RNA polymerase and the rate of something called elongation. Now, I picked this particular question to use in part because the question related to Unit 6, which was one of the units we didn't have time to cover because Minnesota blocked off that 2 weeks after we went into quarantine, and they said no instruction during that time. And I gave some optional work that you could do, but it wasn't graded as part of our class. However, AP is including it. Now, some of you might be like, well, why'd you include it if you didn't teach us about it? And part of it's because I want you to recognize that it will be on the AP test. And also because the kind of funny thing about these AP questions is at first you might look at it and be like, oh, I don't know anything about RNA polymerase. But you can actually earn almost all of the points on this question without knowing anything about RNA. Can't earn them all, but almost all of them. So this is a long FRQ question. This question should have taken you about 25 minutes to answer. And they begin by outlining 2 different experiments. In the first experiment, we see the rate of elongation. Now, if you don't know what that is essentially the rate at which RNA polymerase creates an mRNA strand. The elongation is the elongating of the strand of mRNA. And so we have a wild strain and an experimental strain, which they tell us has a single amino acid substitution. So as I'm reading this question, if I was trying to strategize this as an AP student and this was the question I got, I would immediately start jotting down notes as I read this. So if I read, RNA polymerase, I might just jot a little note for myself like, okay, this is probably going to be about transcription, because that's what RNA polymerase does. If I saw that there was a single amino acid substitution, I might jot down mutations, protein shape, like just the ideas that those things like get me thinking about, so I don't have to go back and reread the whole question. So a little bit you're thinking of strategy as you sort of scan the question. The scientists determine the elongation rate during transcription with and without the modified RNA polymerase enzyme. And then we see a second experiment with a different compound, aminantin, which is a toxic mushroom compound, and it's an RNA polymerase inhibitor, and then they run the experiment again in the presence of aminantin. And we see that they have error bars with 2 standard error of the means. Now, you're not supposed to know anything about amanitin or amanitin. It's an amanita mushroom, amanitin, I don't know. Anyway, you're not supposed to know anything about that. That's the way they're going to ask these questions, is they're going to give you scenarios where you don't know anything about the topic. Exactly. But you can infer a lot of things based on your experience with other topics in the class. All right, so that's the experimental setup. As I was strategizing, I would do a quick scan through, jot down things that stuck out to me, like, oh, they're talking about amino acids, it's about proteins, oh, there's RNA polymerase, it's going to be about transcription and DNA. But I wouldn't spend too much time trying to fully figure out the question or fully figure out the scenario. I move on to the questions and then I go back to the scenario as needed. So question A asked you to describe 3 structural components of an RNA nucleotide monomer. Now, that one does require that you have some background knowledge about the monomers of RNA, of DNA and RNA. We learned about that in trimester 1 when we talked about biochemistry. So an RNA nucleotide is the monomer of RNA and DNA. So nucleotides all have three components. There's a 5-carbon sugar, a phosphate group, and a nitrogen base. Okay, you earned a point if you describe those three structures. Okay, and that what you wrote needs to be written in a complete sentence. So you would need to say something like an RNA nucleotide has a 5-carbon sugar, a phosphate group, and a nitrogen base. Okay. The second point in question A asked you to explain the role of RNA polymerase during transcription. Again, this is a point that's difficult to earn if you didn't do Unit 6 or you don't remember your transcription translation work from last year. But there are two acceptable answers. So you can say that RNA polymerase makes new mRNA molecules based on the DNA template. By matching the DNA base with a complementary base. Or you could have said something like RNA polymerase joins or bonds newly paired RNA nucleotides and the growing R strand with a covalent bond. My guess is most of you would have tried to earn point number 1 on there. So unless you were like thinking about the biochemistry and you're like, how does it physically connect the nucleotides? And you were starting to talk about covalent bonds, you were probably talking about mRNA. It's not enough to just say, though, that RNA polymerase makes mRNA. Okay. So as you're thinking about writing this out, whenever you see the keyword explain, you need to give as complete a description of how that specific process or that's what that specific role is. Now, you don't need to go beyond what RNA polymerase does in transcription, but you do need to explain exactly how does RNA polymerase carry out transcription to earn that point. Okay. If there's questions on that, hop in. Otherwise, I'll move on. Question B was worth 3 points. And here's where being just an adaptable and resilient AP Bio student can help. If you looked at question A and you didn't remember anything about RNA, you might get discouraged and be like, I can't earn any of these points. But question B doesn't require you to know anything about RNA. So identify the independent variable in the experiments. Anytime you have an experiment, the axes can usually tell you the independent and dependent variables. So here on the y-axis, that's almost always the dependent variable. It's the thing that was measured. Okay, so in both of them, it's the maximum elongation rate. And that's what you would need to write as a complete sentence. So you can't just write maximum elongation rate. You'd need to write as a complete sentence, the dependent variable is the maximum elongation rate of mRNA, okay, to earn the point. You need to identify a control group missing from the second experiment. And this question catches students. This one, when this was delivered on the AP test a few years back, very few people earned that point. And the reason they didn't earn that point is because they looked at this experiment and they were like, well, you've got these, so this is your control, so what control is missing? And what they failed to consider is these graphs represent two different experiments. So if you're going to compare the maximum elongation rate in the presence of amanitin solution, you need to know what the elongation rate is without the amanitin solution. So they should have had the wild strain or the experimental strain or both also present in this experiment without the amanitin solution. So that would have been a control. It's not enough that they did the experiment in another trial at a different time. So because the question says in the second experiment, you need to consider only the second experiment on its own. Ignore the fact that they did the first experiment and say, what control are they missing? And here we don't have a control. We don't know. Was there a difference between these two compared to what they would have done normally? We don't know without having the control. So that gets to the third point, which is to justify the need for a control. And your answer should have described that missing a control is needed to interpret the results of experiment number 2. So if either the wild strain or the experimental strain is selected, then you can't. Then you can justify that amanitin has affected the elongation rate. If you don't have that, you can't justify that amanitin has affected the elongation rate. Okay. Question C, describe the effect of amanitin on the maximum elongation rate for the wild type and modified RNA polymerases. Again, this one only requires you to interpret the graph, right? So, so we're trying to talk about what does amanitin do on the wild type and modified, and the answer is it decreases the maximum elongation rate for the wild strain and does not affect the rate in the experimental strain. Again, you would have needed both of those points to earn the point because it asked you to talk both about the wild type and the modified RNA. So you needed to address both. The reason some people get caught on the experimental strain because they're like, it does look like it kind of goes down a little bit. However, if you look at the error bars overlap. So we cannot say there's been an actual decrease. We cannot be confident in that. So if they're just trying to say what happened to the average, because in question C, we're asked to describe on the effect of amanitin on the maximum elongation rate, there is no change. And then the second point on question C was to determine the ratio of the average maximum elongation rate for the modified RNA polymerase compared to the wild strain RNA polymerase in figure 1. So again, this is a question that simply asks you to do some data interpretation. So you're supposed to look at figure 1 and compare and get a difference. So experimental value is an elongation rate of about 2 nucleotides per second, whereas the wild type is 12. So a 2 to 12 ratio comes out to a 1 to 6 or a 1/6 or a 1 to 6 ratio. These are just the answers. If you had done this on the test, you still have to write that as a complete sentence for it to be counted. So the ratio of maximum elongation rate between RNA polymerase and the wild strain RNA polymerase is 1 to 6, would be a complete answer to earn the points. Okay, question D, state the null hypothesis for the experiment in figure 1. So we've done null hypothesis throughout this year. Essentially, it's saying that the independent variable does not affect the dependent variable, but you have to do it for figure 1, okay? So in this one, what is our independent and dependent variable? Our independent variable is experimental strain versus wild strain. So the strain type. Dependent variable is the elongation rate, okay? So the null hypothesis would be any of those. The modified RNA polymerase would not affect the maximum elongation rate. Any difference between the two elongation rates is due to chance. And then you could earn a point for justifying the claim that the change in the amino acid sequence of the modified RNA polymerase affected the shape of the active site. Oops. And so the claim is already there. They've written it out for you. You need to justify it. So you need to provide reasoning and an explanation for why that behavior occurred. So you would have to state something about the active site of the enzyme and that it would decrease the elongation rate. Okay. You could also talk about the change in the shape of the protein structure would change the active site, which would decrease the elongation rate. But it has to mention the active site. Okay. You can't just talk about protein shape being changed by amino acid sequence because they're asking about the active site. Okay. Questions on that one before I move on to the second question? All right. Second question was about pigeons and lice. And if you read this, they talked about how the lice are host specific. They're eating the feathers of the birds. We see here stuff about black and white phenotypes for pigeon color and also for body color of the lice. So immediately I'm already starting to think about phenotypes, lice, they're impacting the pigeons. I'm already thinking about evolution and natural selection, so I would probably jot that down. Then they go on to describe an experiment where lice were transferred from some wild-caught pigeons. So onto groups of black and white pigeons. And then half of the pigeons were fitted with mouthpieces that made it difficult for them to actually remove the lice, which otherwise they would be removing with their beaks. That's what they set up here as they clean their feathers. The lice have a generation time of 24 days. Okay, all right, some other details. And I go on and look at the question. The first one describes the importance of phenotypic variation in louse body color among individuals in the population of lice. You don't have to know anything about lice to answer this. What are they asking? They're asking what's the benefit of physical variation in a population, any population. So this goes back to the basics of natural selection that we've kind of been talking about throughout the year. Which is the idea that variation makes it more likely that some individuals in the population will survive when the environment is changing or when the environment is causing stress to the population. So in this case, the lice, what's their stress? Their stress is pigeons cleaning their feathers and killing the lice. So variation makes it more likely that some of the lice will survive as the color of the pigeons and the pigeons cleaning them change. Okay. Question B, justify the decision. That was worth 1 point, right? You're just saying variation increases the likelihood of survival and reproduction when the environment's changing. Letter B, justify the decision of the researchers to hold lighting conditions constant. This is worth 1 point. This one's a little bit tough because you're going to have to make some inferences or some predictions. You're going to have to be thinking about the experiment sort of holistically. You don't need to know anything about lice and pigeons, but they gave you enough information to be like, well, the pigeons are cleaning the lice off their bodies. So how do you think they find the lice? They probably have to see the lice. So light is going to affect their ability to see the lice. And even if it's not, even if the pigeons are using smell or taste or touch to find the lice that they're cleaning off, it still would be a good idea to control the light conditions on the chance that they're using sight. So you wouldn't want the light to change for different pigeon test groups. Okay. Again, you're just explaining basic experimental design. And then the last two points could be earned for predicting the most likely effect of the pigeons cleaning their feathers on the phenotype of the lice after 4 years. And you're basically predicting what happens as a result of natural selection. So the phenotypes that blend with their host colors would increase in frequency, or the phenotypes that contrast with their host feather colors would decrease in frequency. Either would earn the point. But you have to talk about the phenotypes of the lice. You can't just say light-colored lice will become more common. They're asking about how the phenotypes are going to be affected. So make sure that you're answering the question specifically. Hi, I'll be with you in a second, Zoe. And then provide reasoning to support your prediction for part C. Here's where you can talk about how being a dark colored lice or a light colored lice is an advantage. So lice with contrasting body coloration to the host could be removed before they could reproduce. If you have similar coloration to the host, you're more likely to survive and reproduce. Okay, there's where you can actually talk about the color and which ones are going to survive and reproduce. But notice you have to talk about reproduction. You have to talk about the lice reproducing. That's why certain individuals will become more or less common. So with that, that is a review of the assessment. Does anybody have questions about that? Or does anybody want to share their answers? And hi, Zoe. Everyone's watching you right now. Yeah, they're all there. You just are only looking at their circles. If they turned on their cameras right now, they'd see you. I'm going to stop recording here.